it really is understanding what is the indication that we are trying to target. And we're creating different types of tissue using our fresh 3D bioprinting technology, which allows us to generate tissues of different compositions and essentially infinite geometries, and combining those together to drive the function of these cardiac tissues to then help us pick up on signals early. And so, we really are intent on working together with partners to select these best leads to advance to a preclinical setting. And so, how we do this is we take human cells and extracellular matrix. We are able to build our tissues with full computational design and control of the tissue architecture, as we've previously published. And then use robotic biofabrication, which is 3D bioprinting, and then manipulate our materials in three-dimensional space. And then lastly, turn these tissues into data and readouts that we can actually interpret and pick up on signals. I want to share with you very, very quickly some of the data highlights of, what we've done so far, where we hope to go towards, right? And then this consists in three distinct categories of us, showing that our tissues consist of native-like tissue structure. We have healthy cardiac tissue response, and then that we have the ability to create disease cardiac tissue models, which is really our goal here. So I've shown previously, several different geometries, and we can create rings of different shapes, strips, tissue bands, what we call tissue bands. These are a little bit wider, and then also ventricles. So these are just some examples of different geometries of cardiac tissue that we've been able to fabricate. Of course, the design space is not limited to just these. Each of these have their unique advantages. For example, tissue strips are smaller, easy to manipulate, very consistent. And for a larger tissue band, you start to get a much larger surface area on the tissue and start to be able to pick up more than just a single propagation of calcium signal or contractility. And then for a ventricle, obviously, this is starting to replicate some of the three-dimensional structure and also functional behaviors of a full cardiac tissue. So we build these tissues to address very specific scientific questions, very specific disease phenotypes, and also to help with the analytical readouts that are required. And then within each of these tissues, we really have been able to engineer a very native-like tissue structure. So we have a cell density, final cell density of about 600 million cells per centimeters cubed, which is very, very close to what is in the native adult heart. And then also you can see that the cardiomyocytes are very well aligned, highly aligned in density cellular, which is critical to contractile function. And this is where we think a lot of our capabilities that I'll show later are enabled by. This is just a very basic walkthrough of our analysis. One of our analysis methods of several. Calcium dynamics is critical in understanding some of the disease-modifying effects of compounds. You see on the right here, typically a contractile cycle of cardiomyocytes or the heart goes through a voltage transient leading to a calcium spike, and then leading to the actual physical deformation of the cardiomyocytes themselves that drive a force change, right? And so each of these phases are critical to measure. Here I'm showing that these tissues are contractile, highly contractile, and then also we can collect very high resolution and high sensitivity information by looking at different regions of this tissue. And then we can also start to build other readouts such as conduction velocity that are critical for understanding cardiotoxicity and disease modifying effects, right? It goes from tissue strips where we actually have a very high conduction velocity of about 25 centimeters per second. The adult human heart is at about 40 to 50 centimeters per second. The bands are a little bit smaller and a little bit less, and the ventricles are a little bit less as well. And that's what these bands and ventricle lose out on in terms of their conduction velocity is they actually gain in their amount of calcium propagation information that can be gathered here. And so here, I want to show you some of the actual drug responses. So isoproterenol is traditionally a pretty difficult drug for engineered heart tissues to pick up on, primarily because of the mechanism of action. Isoproterenol is very similar to epinephrine in terms of its effect. It will speed up contractions of the heart and also drive higher contractility, so much larger force generated. So here is a baseline tissue, and then a comparison of that treatment group. You can see it's beating visually faster and also seemingly to deform much more than the baseline. So when we plot these responses, looking at the saturated response as well as the dose response, we see a response very similar to the native myocardium, right? So going from it's speeding up as well as a higher change in amplitude. And this is really starting to prove that our tissues will respond to drugs as anticipated, and that when we are with our disease models, we will be able to take novel compounds and understand their effect against our tissues. I want to show you some examples of different disease models that we can create. One of them being engineered directly from the printed structure itself, right? So this is on screen here. You can see actually several examples of different arrhythmogenic effects. The one on the left here is a multiple wavefront that's propagating within your heart. Your heart typically has a very coordinated stimulation and contraction cycle. And when you have a wavefront, multiple wavefronts occurring, typically what happens is that these wavefronts interfere with the contractility of your heart and interferes with the ability for it to pump, right? And then this eventually leads to cardiac arrest. And then if it happens for too long term, it would turn into heart failure. Another example would be rotors. These pin rotors are a very classical example of arrhythmogenic behavior. And you can actually see another example of that occurring here all the way to the right. So by controlling where we deposit the cells, we can actually control the initiation and propagation of these engineered arrhythmias. And then in a similar vein, we can also start to create some of these fibrotic-like structures that mimic the fibrotic myocardium, right? So fibrosis is a huge problem in not just the heart, but also in every organ system. And usually how fibrosis manifests is in excessive deposition of collagen that surrounds the cells. So you can see in this fibrotic myocardium, the lighter pink is actually where all the collagen surrounds these cardiomyocytes, which should actually natively in healthy hearts, they should be very interconnected with few collagen deposition around. So we can design a construct similar to this fibrotic myocardium and show that the propagation can be controlled based on what we design. And so a lot more complex designs can be engineered here to then move towards, okay, now we have a fibrotic disease model. How do we screen novel compounds that combat the effects of fibrosis, which typically results in arrhythmia, which is not good. And then lastly, we've also started to incorporate a lot of these genetic cells with known genetic mutations, where we're able to start to print them, put them into the tissues, and manifest some disease-like phenotypes in our printed tissues. In this case, is a desmoplakin mutation, which interferes with the cell-cell physical connection between cardiomyocytes. And so some of the keys to building translational disease models that I'll fly through here is, I think, is these 5, right? Better physiology, tunable designs, being able to control our entire process end to end, and then being able to take these tissues, test them against a large variety of compounds, either small molecules, proteins, antibodies, whatever you have, read out the appropriate readouts from each of these to then inform us on how do we select for safer and more efficacious drugs. So, this is how we typically work with the partner, is that we work in a very collaborative process, understanding what is a disease the indication that a customer might care about. What are the key translational readouts that would help a customer identify how the drug of interest affects cardiac physiology, and then we design to purpose, from tissue geometry all the way to its disease modifying effect. So I won't go too much into this, but an example would be, we take a disease like hypertrophic cardiomyopathy, and we then identify, build, validate the model itself, and then select for the compounds that are suitable for further development. So going and screening from 2,000 to 5 compounds, where we're looking at very relevant metrics such as conduction velocity, its contractility, and energetics, which are typically affected in a disease like hypertrophic cardiomyopathy. So that is all I have. Happy to take some questions, and thank you.
Thank you, Andrew. Excellent presentation. You guys made tremendous strides forward. I remember I met your team in 2018, and this is like so much more was done, from not just the bioprinting process, not just the bioprinting product itself, but also commercialization pathway, and also software design. There's just so many things going on. So congratulations. I have a couple questions, and guys, if you have questions, please put them in the QA box. So it looks like we have some of the successful examples of N is equal to 1 at least. What is the process of scale up? Because I would assume that to replace animal models, you will need at least the same number of samples equivalent, to animal models. Like what is your scale up process here?
Yeah, that's a great question. We currently our tissues are printed in 24 well plates, right? And that's where throughput really started to matter, especially when we're looking at screening against 2,000 compounds with a bunch of biological replicates that need to be understood. So we need to eventually move to higher throughput formats. But of course, these tissues will never be as high throughput as what's typically used today, which are 2D monolayer cells, 2D cardiomyocytes on 2D monolayers. So we kind of see ourselves as a medium throughput, somewhere in the medium throughput, but being able to pick up important pieces of information that translates much better to animal models, but importantly to human physiology, right? That would be key, right? Being able to take our assays and translate the results into a human response. And so some of the challenges to scale up would be, I think it's a industry-wide problem, is the amount of cells that are needed. So you can see that I talked about our tissues have a very high cell density, which we think is really what drives our function. But then that also lends itself to a problem where we need a lot of cells to build some of our tissues. And today, iPSC-derived cardiomyocytes are pretty good. There's a very well-established protocol. There are a couple commercial couple companies commercializing iPSC-derived cardiomyocytes, but they are exorbitantly expensive today, right? To produce consistent cell lines, and then also to generate, a mature human-like response. So I think that's one of the places where our industry together will need to work together to improve on, whether it's to scale from, T75s to stacker plates, to then bioreactors, and to then, thousands of liter reactors, and then while maintaining cardiomyocyte function, And then also eventually maturing that cardiomyocyte response is where some of the challenges to scale up happens.
Right. And
Yeah.
actually, at least another question of mine is like, yes, you can produce a functional piece of tissue, but how long do they last? And do you need them to last as long as an animal or some other equivalent can last, like 2D?
Yeah. So a lot of our metrics today are taken at day 14. So 2 weeks out, right? We print our tissues, we keep them in culture for about 2 weeks, and that's very early. And what we are very impressed by is that we're actually able to pick out a lot of the relevant metrics at day 14. Whereas, if you think about how long your maturation process from birth, your heart is constantly developing over the course of years, right? So the tissue needing to last how long, I don't think we really have a clear idea, but we know that today we can pick out a quite a good set of metrics that we can use to screen compounds. But we also have a lot of room for improving, whether it's the maturing by different methods or whether it's keeping tissues in culture for longer. All of that is, I think, all room for improving the tissue and improving the response to drugs.
Right.
So, yeah.
Yeah, and also in pharmaceutical industry, people always shouting, contracting the timeline because it
Yes.
takes so long to figure things out. So maybe it's not a bad thing.
Yeah, and well, compared to animal studies, I think a lot of animal studies actually are challenging because they take so long. Animals take a long time to grow. Mouse obviously not so much, but their gestation period is much shorter. But their response is also not a lot of times not indicative of what a human response might be. And so, what we've heard is that, from some of our partners is they say like, hey, if you can have an assay in vitro, it would save us a lot of time in animal studies where we were just banging our heads against these animals for 1 to 2 years, and then the results might not actually translate towards human response. So, it's definitely an interesting problem, and then also for us to help our partners figure it out.
So we have a couple questions from the audience. I think we have time for one of them, which I think is relevant, and I'm going to modify slightly as well, Andrew. So Prakash asked, how does your technology compare to spheroids or 3D cell cultures? Are you able to obtain more functional tissue? And also, my added on modification in addition to that is, have you guys done a comparison to animal models with your current models?
Yeah, so not yet comparison to animal models, but 100% something that we will be doing is on our roadmap to do, right? That comparison will be critical to validating our technology. The comparisons against organoids, today we compare against 2D monolayers because that's typically what is used to screen different aspects of cardiomyocyte response. And typically, right, it's very apparent that three-dimensional tissue is going to perform better than a 2D monolayer that doesn't consist of any structure. In a 3D organoid is it's closer to a three-dimensional tissue, a little bit closer to what is found in a native heart, just by nature of it having the three-dimensional structure. And it also gives some very interesting responses by how different cell types can reorganize itself. But again, it lacks that sort of alignment, which is critical to cardiac tissues for its contractile function. And so you actually lose out on a lot of information in an organoid structure. So some trade-offs there, of course. 2D and organoids are going to be higher throughput, but the information that we can glean from them is less.
Got it. We have two other questions. They're also very good questions from the audience, but we don't have the time to address. I kind of want to move the webinar forward. So,
Yep.
So Andrew, if you can type in your answers, that would be great.
Yep.
And Move on to the next speaker, Isabella.
Thank you.
Isabella, okay, I'm have to try to pronounce her name. Bundesan, sorry.
That was not too bad, Bundesan, but that's Bundesan is completely fine.
Isabella is a field application scientist from CellLink, which is also a sponsor for our event. Without the sponsor, we will not be able to make this webinar possible. But I know that she has a fantastic presentation coming up as well. So Isabella, I'll let you take over.
Amazing, thank you. I'm just going to start sharing my screen here. Share. There we are. It seems like you all can see it. So that's all good. Great. No, it's exciting to be here today and talking on this important field of drug discovery and drug development. As Jenny mentioned, I'm from CellLink, working as a field application specialist. And what I will talk about here today is really on how 3D bioprinting comes into the picture of drug discovery and drug development. And I do have the luxury in my job of a field application specialist to, on a daily basis, interact with customers and collaborators who are applying bioprinting within these areas, among a lot of other fields. And I will present to you some of these examples where bioprinting is relevant, but also talk a lot about how bioprinting comes into the picture of drug development and why it's so important to incorporate it. And I don't think I need to, or that this slide comes with any news to anyone, as, let me see, I'm just gonna move down the window. As it today is a quite well-established fact that many drugs do fail in the clinical trials, and that we have a great lack of organs for organ transplantation, and that the process of developing new drugs takes way too long time. And the drug development process also costs a lot of money and resources. And this is what really drives us to establish better, more efficient methods to develop new drugs and treatments that are more cost-effective, but also more importantly, more relevant for the patients, so we can get to a faster outcome of the drug result. And this is where bioprinting really comes into the picture, and the 3D cell culturing. And at CellLink, our goal is to be part of the solution for these healthcare challenges, to enable scientific progress by providing technologies, products, and services that supports in this journey. And the vision for most of us working within this field, I believe, is to one day be able to have the capacity and knowledge to recreate organs. But yeah, we still have some way to go to get there. And during this time since bioprinting was established, which I would say were roughly in 2016, it has been discovered that bioprinting is a very versatile technology that can be applied to many different research fields, all the way from the soft robotics and space biology to the more traditional fields in tissue engineering, material development, and drug development. And I would like to highlight some of these applications that plays an essential role specifically for the drug discovery and the drug development area, as that is the topic for the day. And to start off, we have the tissue engineering field. And I would say that this is the base and the space and foundation for the fundamental research, where we can utilize bioprinting to deepen our understanding of biological processes and the biology of the human cells. As bioprinting allows for both multiple cell types, as well as multiple biomaterials, which would serve as extracellular matrix for the cells to come together in a three-dimensional orientation, where both biochemical as well as mechanical cues can be incorporated and tailored to mimic the in vivo environment. Then we have the material development area, which was one of the first established within bioprinting. And I would also say that that is one of the most developed today, as there is an impressive array of smart sensing and reactive materials and biomaterials that are available to use with bioprinting. And these are applied in multiple fields of research. For drug development specifically, it is used to develop biodegradable implants, as well as drug or factor releasing scaffolds, for example, that can be implanted into the body. And if we're looking at the disease modeling, this is an area that is heavily linked to the tissue engineering field, as we already touched upon. But instead of modeling healthy tissue, we are looking to model the diseased tissue. And although this is also a key point to our next area, which is drug testing, where drugs and treatments are tested on in vitro models. And how good the output is of these testings obviously depend on how good of a model we have to test them on. And as it is well established that 3D cell cultures better mimic the native cell conditions, it is critical to have a good disease model, which can recapitulate the intended biological processes to get a faster result on how well a drug or a treatment are performing in the preclinical stages, which we saw excellent examples of just before in the previous talk. And to these models, how much complexity that is brought into these, it can be designed and decided depending on the application. They can be advanced both in terms of personalization by using patient-derived cells, for example, for targeted drug screenings, or in terms of adding additional components such as flow or additional tissue types. So here is where the organ on a chip or multi-tissue models come into place. And something that is not really illustrated here, but that's also very important to touch upon when it comes to personalized medicine, is that bioprinting also can be utilized as a tool to fabricate tailored medication, as bioprinting allows for a small-scale flexible manufacturing of pills. And I will show some examples later on of all these fields. But before that, I won't dwell too much on this slide, as we have talked on a lot of the benefits of using 3D bioprinting. However, I think it's a really good slide to show why 3D cell culturing holds the answer to solve a lot of the obstacles we currently are facing within drug discovery, as the cells gets to interact in a more in vivo-like environment. And then the tools to form these 3D cell culture models are the bioprinters. And there is today a plethora of options when it comes to 3D bioprinters and 3D bioprinting technologies to use. And with these different options, we have the capacity to bioprint on multiple different scales with a lot of different materials with cells embedded into the construct. So we can actually do live cell printing all the way from the micron precision level up to the centimeter and more scale for the large organs. However, there is